Okay, well, good afternoon, everyone. So I'm going to talk about now a little bit more complex systems, and that's, I think, the great thing about Blue Waters, which enables computational research at different levels. I think I'm confident that we're going to hear even larger systems in the next talk, so that's great. So I'm going to talk about actually conformational changes of membrane proteins, and for this purpose, I'm going to use the example of membrane transporters that are sort of pretty important class of membrane proteins. They are all over your body and all organisms, and they are essentially involved in active transport of every species, almost every species that crosses the membrane. You need to have one of these transporters involved in its transport across the membrane. Major target for drugs, we have a lot of them, for example, in your brain, GI, kidney, all over the place. And a main problem in developing new drugs for these transporters is, first of all, we don't know the structure of many of these guys. And secondly, if we have one structure, we can only use that as a target to develop a drug. Whereas if you understand how they move during their function, you might be able to design much more specific drugs for intermediate structures. So a major goal in structural biology of membrane transporters is really to understand how they move during their function. And that's something that we know about these transporters. All these transporters, in order to be able to establish a concentration gradient across the membrane, they have to undergo the so-called alternating access mechanism, which means you can't be open to both sides of the membrane at the same time. If you do that, you dissipate the concentration gradient. You have to grab the substrate from one side, close that side before you open the other side, which means the protein has to undergo very large conformational changes. And that's, of course, I mean, in order to do this, describe this properly, you need to also take into account the chemistry, which is the binding of the substrate, what is the source of energy driving the reaction in a certain direction, which means you have to also include some chemistry details, chemical details. Not at the level that we heard from the previous talk, but you need to have at least atomistic description. So you have very large systems and very complex kind of conformational changes. These are some examples of these transporters, just for you to appreciate that the alternating access mechanism, switching accessibility from the one side of the membrane to the other side, can take place in very different forms. This is, for example, an ATP-driven primary transporter, in which we have the flip-flop motion. This is another example of a sugar transporter coupled to phosphate, in which you also have a flip-flop motion, and this one shows an elevator kind of motion. This is a neurotransmitter transporter, which is responsible for glutamate transporter across the membrane. So very complex, and that's actually a key word here, and that's why we rely on Blue Waters and large computer clusters. In order to describe this complex transition pathway between the two intermediates, as you will see, sometimes you have to take several stages and steps. And that's not possible to do using a simple reaction coordinate, and that's why we have to do a very comprehensive search before we can go on and do free energy calculations and sort of more detailed description of the system. So let me actually give you some example and just talk about one method one new approach that we developed in order to deal with these proteins. First of all, I think it's pretty clear that we have to resort to non-equilibrium methods. If you do equilibrium calculation on these proteins, nothing is going to happen within the time scales that we can simulate, which is nowadays on the order of microseconds or so. We have to do non-equilibrium. So let me give you a couple of examples in which we think we have made significant progress. The first example, we are taking advantage of a lucky transporter for which we have 3 experimentally characterized structures. So we know that during the function, it has to go from one state to the other, probably visiting this intermediate state. And just to test our approach, we want to describe this transition, but of course, we have no idea of the pathway. And as you can imagine, in the multidimensional space, there are millions of ways or infinite number of ways of going from A to B. The question is, which one is the correct pathway? Equilibrium simulations, if you start from any of these states, they essentially stay where they are. OF stands for outward facing, IF stands for inward facing, and you would like to be able to see the transition between these two states. But of course, this is a very slow process, a millisecond process, so you're stuck here if you just do routine equilibrium simulation. You don't see the transition. So we have to do non-equilibrium calculations. But again, along what pathway? That's the key question here. So this is just to describe the problem in very simple terms. I have been using this first time in Telluride when I gave this talk. So you want to go from one valley, Telluride, to the other valley, mountain village. And then it's probably sort of, you have no idea of the energy landscape here. It's at night and you don't see anything. And of course, I mean, you just want to go using sort of some interpolation or conventional methods using a simple reaction coordinate like RMSD. You can have a sort of almost linear connection between the two, which might take you over a very large barrier, unnaturally and unphysically. That would be your actual reaction pathway, right? Because this is along the valleys. If you start with this, then you can actually sort of try to refine this using advanced sampling methods and computational methods, but it's not going to take you all the way to the actual reaction pathway, which is something like this in this particular case. And please remember, again, I'm trying to make a point that this is a very complex molecular transition. It's not one transition. It goes probably from here to there, and then go around and come here. It's a multi-stage process. But we don't have enough experimental information to be able to connect these dots. The only structures that we have are these two. In this particular case, we have a third one that we use to test our method, but most of the time you have only one structure actually, sometimes two. So, and again, as I pointed out, usually you have no notion of the background energy landscape and you want to describe this complex problem computationally. So, in this particular case, actually, if you do TMD, targeted MD, that uses a very convenient reaction coordinate, RMSD in this case, you convert this transporter to a channel in the middle, which we know from a mechanistic perspective that is also incorrect. So, but again, I mean, we have developed a method to look at this more quantitatively for any protein in which you're interested in in large-scale conformational changes. So again, this is the problem now. The first sort of novel aspect of our approach is, instead of actually taking a bad initial guess, and again, I would like to emphasize, we do TMD for simpler systems in which the reaction coordinate is pretty clear, but if your system is complex, TMD usually fails, as I showed you. Instead of using a sort of bad initial guess and spend a lot of time refining it, so we suggest that maybe we should try more and explore this landscape many more time, and that's actually, we spent at least half of our computational effort on this step in which we try to explore this space. This is a very huge space, and we run on the order of hundreds of different initial exploration calculations to see, to explore this space. And then the second part is somehow you have to be able to compare the quality of these potential initial guesses, and for that we use non-equilibrium work. So these are the two sort of novel aspects here. So we try to define relevant reaction coordinates instead of using computationally convenient reaction coordinate. We try to look at the protein, becomes very empirical in a way. So what are the global degrees of freedom that are changing, going from one step to another one, and try to align our collective variables along those degrees of freedom. That's one thing. And then we do a lot of quick simulation, 20, 50, 100 nanosecond, and you can appreciate these are very easy to parallelize. But we have to do a lot of them. And then we compare these quick simulations by looking at the non-equilibrium work, how much work it takes to take the system from state A to B through very quick simulations when you push the system. And then by doing this and repeating this calculation, you start seeing actually patterns of what reaction coordinate might be more comfortable for the protein, and the protein is more compliant, and it's potentially a better initial guess for our more expensive calculation. Then we take those calculations, and again, we do free energy calculation using multi-dimensional Hamiltonian replica exchange, and that's really the expensive part that was, for these complex systems, impossible before we had machines like Blue Waters and some of the DOE machines. And of course, at the end, again, you have to look at this pathway and make sure it makes sense, it's correct, The molecule is doing well, mechanistically it makes sense, it's consistent with experiment. So that's something that sometimes some colleagues miss, and the numbers look right when you look at the transition. It's something is fundamentally wrong. So, okay, so in terms of technical aspects, so now instead of using RMSD for this time-dependent, excuse me, potential that we have here, instead of using a RMSD, which is conveniently implemented in all programs that we have for simulations, we can have a variety of different, what we call collective variables, combine them linearly. They have been implemented in NAMD, and we are developing also a tutorial for them for people who are interested in using them. So, and then we push the system along many different pathways. So that's one example. For example, for this transporter, we know that we have this transition of one domain with respect to the other domain. So we can describe this transition as a combination of a translation and a rotational degree of freedom. But of course, the question is whether the translation happens first and then rotation, or it's stepwise. You can imagine many, many possible pathways, including these two reaction coordinates. But now we can actually use Blue Waters, have 200, 300 parallel simulations in which we try all possible transitions and then compare the work for these transitions using non-equilibrium work and take the best one, do a slower simulation, and then once we start converging, then we take that one, do Hamiltonian replica exchange to calculate the free energy, which is the ultimate proof for the quality of that pathway. So then I'll just show you a couple of examples. So this is one example of a transporter. In this case, we are using, for example, opening, closing on one side, opening, closing on the other side, the twist angle between some of the domains and their combination, different force constants. And in this case, we are looking at 200 independent initial non-equilibrium trajectories. So these are all sort of shown in this space. And you can see they are taking different mechanisms. This is actually just a handful of them shown here. Take different mechanisms. In some cases, beta changes before gamma. In some cases, you have different mechanisms here. But we can really sort of steer the system along any pathway that we think might be relevant to the mechanism. And then we compare, of course, the work here. And you can, this is really the key part here. So different mechanisms. And by just comparing the work, how much effort it takes to push the protein along certain pathways, you can see there is a huge difference between the red line is usually TMD in all my graphs, the conventional way of pushing the method. You need more than 500 kilocalorie per mole to induce the transition in 20 nanoseconds. But by designing a better collective variable that makes more sense, it's a little bit more difficult, takes more effort, but it becomes much easier for the protein to comply and to undergo the conformational change. And then, of course, then you can actually, even in this case, and this is another key aspect here, you start actually looking, by just looking at these 200 different ways that you push the protein, you start seeing clusters of work. For example, you see every time you work with alpha before beta, then the work value is high, meaning that the protein doesn't like to open this end before it closes the other end, which also makes mechanistically sense. So you start noticing actually that there seems to be a sequence, an order of events involved. It's not a simple process. There are many steps involved, and you have to take this into account. And sort of naturally, from your comprehensive search comes out the sort of more potential and more reasonable mechanism in which D changes before beta, before gamma, before alpha. So this is actually based on using that reaction sort of set of transitions. So this complex reaction can be described here. And again, I would like to point out that here we have all the simulations are atomistic. We have explicit representation of membrane, water. These are on the order of 200 to 300,000 atom systems. So every copy of the simulation is already large, and we need to have many, many copies of these to do the free energy calculation, which is what I'm going to show next. So for this particular case, for example, we came up with the concept of doorknob mechanism. These two parts, yellow and green, are very difficult to separate unless you twist them with respect to each other like a doorknob before you open it. And that comes out very nicely from our work value, but also from our free energy calculations. So, very good. So now, I mean, we can also do free energy calculations. Here we use replica exchange. In this particular case, we use 22 replicas. And again, that's the part that can easily get very, very expensive. Very nice convergence for those of you who are interested in the details. And then comes out the free energy, for example, along the opening of the alpha in this particular case. So what we are showing here is that, what we are suggesting here is that one of the crystal structures corresponds to a real free energy coming out of our calculations, whereas the other crystal structure doesn't seem to be a minimum energy, probably the result of crystal contact, which was also proposed by other experiments. We think this is the real sort of minimum energy. So for the next example, let me actually just run. We are using the same approach here in this case. It's a different transport, a different kind of transition. Again, we see a very reasonable, mechanistically consistent picture of the transition between the two states using very simple reaction coordinates after doing a sort of extensive search. And then we, again, using even before we go to free energy calculation, using non-equilibrium method here, we can actually see the effect of substrate binding. Substrate binding makes it easier for the protein to undergo the conformational change between the two states. But the point here is that now we understand how the protein moves, and now we want to add the effect of the substrate, another dimension to our complexity. We already needed several degrees of freedom combined to describe the protein, and now you have to throw in the substrate and see where the chemistry takes place and how the chemistry is going to affect. So adding the substrate suddenly makes it much more complex. So you start dealing with multidimensional space. Let me actually skip this one. Just to show you again the kind of calculations that we need. We have to go through several iterations across multiple axes here. And then after each iteration of replica exchange, Hamiltonian replica exchange calculation, we have to optimize the position of these and then go through several steps. Sometimes you have to do two-dimensional replica exchange calculation. And then at the end, we put everything together, for example, using 150 replicas, to look at the entire transition and make sure that we have a real transition between all these points. That becomes really expensive and only possible on resources like Blue Waters. So when you do this, so this is what I claim the first time you see a free energy profile for the entire transition of this protein without the substrate is very difficult. High free energy, more than 15 kilocalorie per mole. You have the substrate binding here, and then in the presence of the substrate, the free energy against transition comes down to 10, which matches very nicely, very close to what we know from experiment. And you have included all the details in this free energy. That's just to show very interestingly, actually, that the transition between the bound state and unbound state for different IF state and inward-facing state and outward-facing state is different. You go like that to the other state, and you come back through a different pathway. So there are two different pathways involved in the presence of the substrate and in the absence of the substrate. And this is going to be my last slide, just to emphasize, this took us more than 1.5 years to take into account the effect of the substrate and rearrangement of the substrate binding site. Really, without describing this properly, we couldn't get this thing right. So which means that there's just one example of the complexity that you have to deal with when you're working with these complex monsters, essentially. All right, well, I thank my group, great collaborators, and of course, computational resources, without which we can't do any of this. Thank you very much for your attention.